Chapter 14, The Cardiovascular System. This chapter focuses on the fundamental concepts of cardiovascular physiology, divided into four key sections. The first section provides an overview of the cardiovascular system, covering its major components, including the heart, blood vessels, and blood, and explaining how they work together to transport essential materials throughout the body. The second section explores pressure, volume, flow, and resistance, examining the physical principles that govern blood movement and distribution. It explains how pressure gradients drive blood flow and how vascular resistance influences circulation. The third section delves into cardiac muscle and the heart, focusing on the specialized structure and function of cardiac muscle cells. This section also covers the mechanisms of electrical activity, contraction, and the unique properties of the heart that allow it to function as a continuously active pump. The final section explains the heart as a pump, detailing the cardiac cycle, heart sounds, and how the coordinated contraction and relaxation of heart chambers ensure efficient circulation. By the end of this chapter, you will have a comprehensive understanding of the cardiovascular system's function and regulation, setting the foundation for more advanced topics in cardiovascular physiology. The cardiovascular system is essential for sustaining life, functioning as the body's transport network. It consists of three key components: the heart, which acts as a pump, blood, which serves as the fluid medium, and blood vessels, including capillaries, which form the intricate tubing system that circulates blood throughout the body. Beyond circulation, the cardiovascular system plays a crucial role in transporting vital materials. It brings in nutrients, water, and gases from the external environment, distributes hormones, immune cells, and antibodies between tissues, and removes waste products like carbon dioxide, heat, and metabolic byproducts. This dynamic system ensures that every cell in the body receives the substances it needs while efficiently eliminating waste. The cardiovascular system is composed of three main components: blood vessels, the heart, and blood itself. Together, they work to transport oxygen, nutrients, and waste products throughout the body. Blood vessels, also known as the vasculature, include arteries, veins, and capillaries. Arteries carry blood away from the heart, while veins return blood back. Capillaries are the site of exchange, where oxygen and nutrients diffuse into tissues. Additionally, the portal system connects two capillary beds in series, facilitating direct communication between organs. The heart, a muscular organ, is divided into two halves by the septum, the left and right sides. Blood returning to the heart enters the atrium, while the ventricles pump blood out, ensuring continuous circulation. Blood itself is composed of cells and plasma, playing a critical role in carrying oxygen, immune cells, and waste products. Finally, circulation can be divided into two main loops: pulmonary and systemic. Pulmonary circulation involves the movement of blood between the heart and lungs, distinguishing between pulmonary arteries and veins. Systemic circulation delivers oxygenated blood to the rest of the body, with the aorta handling cardiac output and the vena cava managing venous return. We'll explore this in more detail in the next slide. This diagram provides a comprehensive overview of the cardiovascular system, illustrating the interconnected network of blood vessels that transport oxygenated and deoxygenated blood throughout the body. At the center of circulation is the heart, which pumps blood through two major circuits: the pulmonary and systemic circulation. Deoxygenated blood from the body returns to the right atrium via the superior and inferior vena cava. It then moves into the right ventricle, which pumps it to the lungs through the pulmonary arteries. In the lungs, gas exchange occurs, allowing the blood to release carbon dioxide and absorb oxygen. Oxygenated blood then travels back to the heart via the pulmonary veins, entering the left atrium. From here, it moves into the left ventricle, which pumps it out through the aorta. The aorta branches into major arteries, supplying oxygen-rich blood to the head, arms, digestive tract, liver, kidneys, and lower extremities. The diagram also highlights the portal system, where blood flows between the digestive tract and the liver via the hepatic portal vein. Additionally, the renal arteries and veins ensure continuous filtration of blood by the kidneys. Together, these components maintain circulation, delivering essential nutrients and removing metabolic waste, ensuring the body's tissues receive the support they need to function efficiently. Blood circulation is driven by pressure differences within the cardiovascular system. Blood flows from regions of high pressure to regions of low pressure, ensuring continuous movement through the vessels. This movement is governed by a pressure gradient, represented as delta P, which refers to the difference in pressure between 2 points in the circulatory system. The greater the difference, the stronger the driving force for blood flow. The heart generates the highest pressure as it contracts, propelling blood into the arteries. As blood moves through the circulatory network, pressure gradually decreases, allowing it to return to the heart through veins, which operate at a much lower pressure. Blood pressure is a key measure of hydrostatic pressure, the force exerted by blood against the walls of blood vessels. This pressure is essential for maintaining circulation and ensuring that organs receive an adequate supply of oxygen and nutrients. Blood circulation is driven by a pressure gradient, where blood moves from areas of higher pressure to areas of lower pressure. This graph illustrates the decline in mean systemic blood pressure as blood flows from the aorta through the systemic circulation. At the start of systemic circulation, the highest pressure is observed in the aorta, with an average of 93 millimeters of mercury. As blood moves into the arteries and arterioles, pressure gradually decreases due to the branching and increased resistance within these vessels. The most significant drop in pressure occurs at the level of the arterioles, which regulate blood flow into the capillaries through vasoconstriction and vasodilation. By the time blood reaches the capillaries, pressure has already decreased substantially, allowing for the exchange of gases, nutrients, and waste. As blood moves into the venules, veins, and ultimately the vena cava, pressure continues to drop, reaching just a few millimeters of mercury. This low-pressure environment allows blood to return to the heart, where the cycle begins again. This pressure gradient is essential for maintaining continuous circulation, ensuring that oxygen and nutrients are delivered efficiently to tissues while waste products are removed. Blood pressure regulation is influenced by changes in blood vessel diameter rather than changes in blood volume. This principle is essential for maintaining proper circulation and ensuring that organs receive the oxygen and nutrients they need. In blood vessels, dilation or widening of the vessel decreases blood pressure, allowing for increased blood flow. Conversely, constriction or narrowing of the vessel increases blood pressure by restricting blood flow and increasing resistance. The cardiovascular system uses 2 key mechanisms to regulate blood pressure: vasoconstriction and vasodilation. Vasoconstriction increases blood pressure by reducing vessel diameter, while vasodilation decreases blood pressure by expanding the vessel lumen. Ultimately, blood flow is governed by the pressure gradient. Fluid moves from areas of higher pressure to areas of lower pressure, and the greater the pressure difference, the faster the blood flows. This ensures a steady and efficient delivery of blood to all tissues in the body. Blood flow within the cardiovascular system follows fundamental principles of fluid dynamics. One key concept is that fluid only moves when there is a pressure gradient, meaning a difference in pressure between 2 points. In the diagram on the left, we see that fluid flows from an area of higher pressure to an area of lower pressure. However, if the pressure is equal throughout the tube, no flow occurs because there is no driving force. The diagram on the right demonstrates another important principle. Flow is determined by the pressure gradient, not by absolute pressure. Even though the pressures in the two tubes are different, the pressure gradient in both cases is the same, 25 millimeters of mercury. As a result, flow remains equal in both tubes. This principle is essential in cardiovascular physiology. It explains why blood continues to flow efficiently throughout the body, regardless of absolute pressures, as long as a pressure difference exists to drive circulation. Blood flow in the cardiovascular system is influenced by resistance, which acts as an opposing force to circulation. The relationship between flow and resistance is inverse, meaning that as resistance increases, flow decreases. Several factors contribute to resistance within blood vessels. First, resistance is directly proportional to the length of the vessel. Longer vessels create more friction, increasing resistance. This is why obesity and excessive tissue growth can contribute to higher blood pressure. Second, resistance is also proportional to the viscosity or thickness of blood. The thicker the blood, the harder it is to push through vessels, increasing resistance. Conditions such as dehydration or elevated red blood cell counts can make blood more viscous and reduce flow. The most significant factor affecting resistance is vessel radius. Resistance is inversely proportional to the fourth power of the radius, meaning that even small changes in blood vessel diameter can have a dramatic impact on flow. Vasoconstriction, or narrowing of blood vessels, increases resistance and reduces flow, while vasodilation, or widening of vessels, decreases resistance and improves circulation. Ultimately, blood flow depends on both the pressure gradient and resistance. Flow increases when the pressure gradient rises, and it decreases when resistance to flow is high. This dynamic regulation ensures that blood reaches the tissues that need it most, adapting to the body's ever-changing demands. Mean arterial pressure, or MAP, is a key measure of overall blood pressure regulation. It represents the average arterial pressure throughout one complete cardiac cycle, including both systole, the contraction phase, and diastole, the relaxation phase. MAP is primarily influenced by two factors: cardiac output and peripheral resistance. Cardiac output refers to the total volume of blood ejected by the ventricles per minute, while peripheral resistance describes the resistance blood encounters as it flows through the systemic circulation. The relationship between these variables is expressed mathematically. Mean arterial pressure is directly proportional to cardiac output multiplied by peripheral resistance. This means that any increase in either cardiac output or resistance will result in a higher MAP, while a decrease in these factors will lower MAP. Understanding MAP is crucial in clinical settings as it provides insight into tissue perfusion and overall cardiovascular function. Maintaining an optimal MAP ensures that organs receive adequate blood flow, supporting their essential functions. The heart is a muscular organ that serves as the central pump of the cardiovascular system. It is divided into 4 chambers, each playing a crucial role in circulation. The upper chambers, known as the paired atria, have thin walls and receive blood returning to the heart. The lower chambers, called the paired ventricles, are thick-walled and responsible for pumping blood out of the heart to the lungs and the rest of the body. At the base of the heart, major blood vessels emerge to facilitate circulation. The aorta and pulmonary trunk carry blood away from the heart, while the vena cava and pulmonary veins return blood to the heart. The heart operates through 2 circulatory loops, one for deoxygenated blood and one for oxygenated blood. Deoxygenated blood from the body enters the right atrium through the vena cava. It then moves into the right ventricle, which pumps it into the pulmonary trunk and toward the lungs for oxygenation. Once oxygenated, blood returns to the heart through the pulmonary veins, entering the left atrium. It then moves into the left ventricle, which pumps it into the aorta for distribution throughout the body. This continuous cycle ensures that oxygen-rich blood reaches tissues while carbon dioxide is carried away for removal through respiration. The heart is centrally located within the thoracic cavity, specifically within a region called the mediastinum. It is positioned on the ventral side of the thorax, nestled between the lungs and above the diaphragm. Several anatomical structures surround and support the heart. The trachea, or windpipe, lies superior to the heart and serves as the airway for respiration. The thyroid gland, located near the trachea, plays a role in hormone regulation and metabolism. The first rib, shown cut in this diagram, marks the upper boundary of the thoracic cavity, while the diaphragm forms the lower boundary, acting as a muscular partition between the thoracic and abdominal cavities. The heart itself has an apex, which is the pointed lower end that tilts slightly to the left. This orientation allows for efficient blood ejection and ensures that circulation is properly directed through the major blood vessels emerging from the base of the heart. By being centrally located and well protected by the rib cage, the heart is optimally positioned to pump blood effectively throughout the body, ensuring continuous circulation and oxygen delivery to tissues. The heart is enclosed within a protective, fluid-filled sac known as the pericardium. This membranous sac reduces friction as the heart beats and provides a cushioning layer to prevent excessive movement within the thoracic cavity. In the detailed anatomical view, we see that the ventricles make up the bulk of the heart structure. These thick-walled chambers are responsible for pumping blood forcefully into the systemic and pulmonary circulations. Several major blood vessels attach to the base of the heart, ensuring the efficient transport of blood. The aorta, the body's largest artery, carries oxygenated blood from the left ventricle to the rest of the body. The pulmonary artery, in contrast, carries deoxygenated blood from the right ventricle to the lungs for gas exchange. The superior vena cava returns deoxygenated blood from the upper body to the right atrium, while the coronary arteries and veins, located on the surface of the heart, provide essential circulation to the heart muscle itself, ensuring it receives adequate oxygen and nutrients. Together, these structural features allow the heart to function as a powerful, efficient pump, maintaining continuous circulation and supporting life-sustaining processes throughout the body. The heart maintains efficient circulation through a system of one-way valves, ensuring that blood flows in the correct direction with each heartbeat. In this detailed anatomical view, we see the atrioventricular valves, also known as the tricuspid and bicuspid or mitral valves, which separate the atria from the ventricles. These valves prevent the backflow of blood when the ventricles contract. The chordae tendineae, often referred to as heartstrings, anchor these valves to the papillary muscles, providing additional support during contraction. At the base of the pulmonary artery and aorta, the semilunar valves, specifically the pulmonary and aortic valves, prevent blood from flowing backward into the heart after it has been ejected into circulation. These valves play a crucial role in maintaining unidirectional blood flow through the cardiovascular system. The inset image highlights the unique structure of myocardial, or cardiac muscle, cells. Unlike skeletal muscle, myocardial cells are branched and contain a single nucleus. They are connected by specialized junctions known as intercalated discs. These discs allow electrical impulses to travel rapidly between cells, enabling coordinated contractions that produce the rhythmic beating of the heart. Together, The heart's valves and specialized muscle cells ensure efficient pumping, regulating blood flow and maintaining circulation throughout the body. This table provides a summary of the heart's chambers and major blood vessels, highlighting the pathways through which blood circulates. The blue text indicates structures carrying deoxygenated blood, while the red text represents structures carrying oxygenated blood. In the heart, the right atrium receives deoxygenated blood from the vena cava and sends it to the right ventricle. The right ventricle then pumps this blood to the lungs through the pulmonary trunk, where gas exchange occurs. Once oxygenated, blood returns to the heart through the pulmonary veins, entering the left atrium. From there, it moves into the left ventricle, which pumps oxygen-rich blood into the aorta for distribution to the body. The table also outlines major blood vessels involved in circulation. The vena cava collect deoxygenated blood from the systemic veins and deliver it to the right atrium. The pulmonary trunk carries blood from the right ventricle to the lungs, while the pulmonary veins return oxygenated blood to the left atrium. Finally, the aorta receives blood from the left ventricle and distributes it to systemic arteries, ensuring that all organs receive the oxygen and nutrients they need. Together, these structures maintain a continuous and efficient flow of blood, supporting vital functions throughout the body. To maintain efficient circulation, the heart relies on a system of valves that ensure one-way blood flow, preventing backflow and maintaining proper pressure dynamics. There are two main types of heart valves: atrioventricular valves, or AV valves, and semilunar valves. The atrioventricular valves separate the atria from the ventricles and prevent the backflow of blood during ventricular contraction. These valves are supported by chordae tendineae, which anchor the valve flaps to papillary muscles. This structure prevents the valves from everting or flipping backward into the atria when the ventricles contract. The tricuspid valve, located on the right side of the heart, controls blood flow between the right atrium and right ventricle. On the left side, the bicuspid valve, also known as the mitral valve, regulates blood flow between the left atrium and left ventricle. Semilunar valves, on the other hand, are positioned between the ventricles and major arteries. The aortic valve ensures that blood flows from the left ventricle into the aorta, preventing backflow into the heart. Similarly, the pulmonary valve regulates blood flow from the right ventricle into the pulmonary trunk, directing deoxygenated blood to the lungs. Together, these valves work in coordination to ensure that blood moves efficiently through the heart and into the systemic and pulmonary circulations, supporting the body's oxygen and nutrient demands. The heart's valves play a crucial role in maintaining unidirectional blood flow and preventing backflow during the cardiac cycle. This diagram highlights the function of the atrioventricular (AV) and semilunar valves during ventricular contraction. In the frontal section, we can see the left AV valve, also known as the mitral valve, positioned between the left atrium and left ventricle. During ventricular contraction, the AV valves remain closed, ensuring that blood is ejected forward into the arteries rather than flowing backward into the atria. The chordae tendineae, which are tense during contraction, prevent the valve flaps from everting under the force of high pressure. The transverse section B provides a different perspective, showing both AV valves from the atrial view and the semilunar valves from inside the aorta and pulmonary artery. Here, the mitral and tricuspid valves are tightly closed, while the aortic and pulmonary semilunar valves remain open, allowing blood to exit the ventricles and enter the major arteries. This coordinated valve function ensures that blood moves efficiently through the heart and into systemic and pulmonary circulation, supporting continuous and controlled blood flow. During ventricular relaxation, or diastole, the heart's valves coordinate to allow blood to fill the ventricles while preventing backflow into the heart. In the frontal section, C, we see that the left ventricle is dilated as it relaxes, allowing blood to enter. The atrioventricular AV valves, such as the mitral valve, are open, permitting blood flow from the atria into the ventricles. Meanwhile, the chordae tendineae and papillary muscles are relaxed, ensuring that the AV valves remain open during this phase. The semilunar valves, including the aortic and pulmonary valves, are closed during relaxation. This prevents blood that has been ejected into the arteries from flowing back into the heart, maintaining forward circulation. In the transverse section, D, we see a top-down view of the heart, showing both AV valves open and the semilunar valves closed. This visualization reinforces the importance of valve function in directing blood flow efficiently through the cardiac cycle. This cycle of valve opening and closing ensures that blood moves in only one direction, allowing the heart to effectively pump oxygenated and deoxygenated blood throughout the body. The coronary circulation is responsible for supplying oxygen-rich blood to the heart muscle itself, ensuring that it has the necessary nutrients and oxygen to function effectively. It consists of a network of arteries and veins that supply and drain blood from the heart's surface. The left side of the diagram shows the anterior view of the heart. The left and right coronary arteries branch off from the aorta just above the aortic valve. The left coronary artery, or LCA, further divides into the circumflex branch and the anterior interventricular branch, also known as the left anterior descending artery, or LAD. These arteries deliver oxygenated blood to the left atrium, left ventricle, and portions of the right ventricle. The right coronary artery, or RCA, supplies blood primarily to the right atrium and right ventricle. It also contributes to the posterior circulation of the heart. On the right side of the diagram, we see the posterior view of the heart. The coronary sinus, a large venous structure, collects deoxygenated blood from the great cardiac vein, middle cardiac vein, and small cardiac vein. This blood is then returned to the right atrium, where it will be sent to the lungs for reoxygenation. By maintaining continuous circulation through this specialized network, the heart ensures that its own muscular tissue remains well oxygenated and capable of sustaining its essential pumping function. Cardiac muscle is composed of two main types of cells: autorhythmic cells and contractile cells. Each plays a distinct role in maintaining the heart's rhythmic contractions and efficient pumping function. Autorhythmic cells, also known as pacemaker cells, are specialized to initiate and regulate the heart's electrical activity. A key example is the sinoatrial, or SA, node, which generates the cardiac action potential. These cells are smaller than contractile cells and contain fewer contractile fibers. Unlike contractile cells, they do not have organized sarcomeres, meaning they are not responsible for forceful contraction, but instead coordinate the heart's rhythmic beating. Contractile cells, on the other hand, are responsible for generating the force needed to pump blood. They contain striated fibers that are organized into sarcomeres, similar to skeletal muscle, allowing them to produce strong, coordinated contractions. When comparing cardiac muscle to skeletal muscle, there are several key differences. Cardiac muscle fibers are smaller and contain only a single nucleus per fiber. They also have a branching structure that allows them to form a connected network with neighboring cells through specialized intercalated discs. These intercalated discs contain gap junctions, which facilitate electrical communication, and desmosomes, which provide structural stability by holding cells together during contraction. Together, these cellular components ensure that the heart functions as a synchronized, efficient pump, maintaining continuous circulation throughout the body. Cardiac muscle is uniquely structured to support the heart's continuous pumping action. This diagram highlights two key features of cardiac muscle: its spiral arrangement and its intercalated discs. In the left image, we see that ventricular muscle fibers are arranged in a spiral pattern. This organization allows the heart to contract in a twisting motion, squeezing blood upward from the apex toward the base of the heart. This movement ensures efficient ejection of blood into the major arteries. On the right, a close-up view of cardiac muscle cells reveals the specialized structures that facilitate contraction and communication. Intercalated discs, located between cardiac muscle cells, contain desmosomes and gap junctions. Desmosomes help transfer mechanical force from one cell to another, preventing cells from separating during contraction. Meanwhile, gap junctions allow for the rapid passage of electrical signals, ensuring that cardiac muscle cells contract in a synchronized manner. Each cardiac muscle cell contains a single nucleus and numerous mitochondria, which provide the energy needed for continuous contraction. Contractile fibers within these cells enable force generation, allowing the heart to function as an efficient pump. Together, these features ensure that cardiac muscle operates as a cohesive unit, allowing the heart to maintain a steady and effective circulation of blood throughout the body. Unlike skeletal muscle, cardiac muscle contraction is graded, meaning the strength of contraction can vary depending on physiological needs. Two main factors determine the force of contraction: the number of active cross bridges and sarcomere length. The number of active cross bridges is directly proportional to the amount of calcium bound to troponin. A higher calcium concentration results in more cross-bridge formation, leading to a stronger contraction. This calcium-dependent regulation allows the heart to adjust its force of contraction based on demand, such as during exercise or stress. Additionally, sarcomere length influences the force of contraction through the length-tension relationship. An optimal sarcomere length ensures maximal cross-bridge formation, generating the highest contractile force. However, if the sarcomeres become excessively stretched, the ability to generate force is reduced. A clinical example of this principle is dilated cardiomyopathy, a condition in which the heart's ventricles become enlarged and weaken. As the heart muscle stretches beyond its optimal length, contractile force decreases, leading to impaired pumping efficiency and heart failure over time. By regulating calcium availability and maintaining optimal sarcomere length, the heart ensures that contraction strength adapts to meet the body's circulatory demands. The electrical activity of myocardial contractile cells follows a distinct action potential pattern, which ensures effective contraction and prevents sustained muscle contraction, known as tetanus. This process occurs in 5 phases, each characterized by specific ion movements. In phase 4, the cell is at its resting membrane potential, maintaining a stable voltage until an action potential is triggered. Phase 0, or depolarization, begins when sodium voltage-gated channels open, allowing a rapid influx of sodium ions into the cell. This causes the membrane potential to become more positive. At the same time, slow potassium and calcium channels start to open. In phase 1, initial repolarization occurs as sodium channels close. A different type of potassium channel briefly opens, allowing a small amount of potassium to exit the cell. These fast potassium channels then close, leading to the next phase. Phase 2 is known as the plateau phase. During this time, slow calcium channels fully open, allowing a steady influx of calcium ions. This prolonged depolarization is crucial because it sustains the refractory period, preventing the heart muscle from experiencing sustained contraction and ensuring proper rhythmic contractions. Finally, phase 3 marks rapid repolarization. Potassium exits the cell through slow potassium channels, restoring the cell's resting membrane potential and preparing it for the next action potential cycle. This unique action potential pattern in cardiac muscle cells ensures control, rhythmic contractions that maintain efficient blood circulation throughout the body. The action potential of a cardiac contractile cell follows a unique sequence of ion movements that ensures controlled contraction and relaxation of the heart muscle. This graph illustrates the different phases of the action potential, along with the corresponding ion channel activity. The process begins with phase 0, where sodium channels open, allowing a rapid influx of sodium ions into the cell. This causes a sharp depolarization, bringing the membrane potential to a more positive value. In phase 1, sodium channels close, and a brief repolarization occurs as fast potassium channels open, allowing potassium to exit the cell. Phase 2, known as the plateau phase, is marked by the opening of slow calcium channels, allowing calcium ions to enter the cell. This calcium influx helps sustain the contraction and prolongs depolarization. Meanwhile, some fast potassium channels remain open, balancing the charge. In phase 3, calcium channels close while slow potassium channels open. This allows potassium ions to exit the cell, leading to repolarization and bringing the membrane potential back toward its resting state. Finally, phase 4 represents the resting membrane potential, where the cell returns to its baseline state, ready for the next action potential cycle. This sequence of ion channel activity ensures that cardiac muscle cells contract and relax in a coordinated manner, maintaining the heart's rhythmic pumping action. The refractory period in cardiac muscle is a critical feature that ensures proper heart function by preventing sustained contractions, also known as tetanus. In the left graph, we see that in a cardiac muscle fiber, the refractory period lasts almost as long as the entire muscle twitch. This means that the heart muscle cannot be re-stimulated until the previous contraction is nearly complete. This extended refractory period ensures that the heart relaxes between beats, allowing for proper filling of the chambers before the next contraction. In contrast, the right graph illustrates how skeletal muscle behaves when the refractory period is short. In skeletal muscle, repeated stimuli can lead to summation, where contractions build upon each other, and, in extreme cases, tetanus occurs. However, in cardiac muscle, the long refractory period prevents this from happening. The heart cannot enter tetanus because it must continuously alternate between contraction and relaxation to maintain effective blood circulation. This fundamental difference between cardiac and skeletal muscle is crucial for sustaining life, as it allows the heart to beat rhythmically without prolonged, uncoordinated contractions. Unlike cardiac muscle, skeletal muscle fibers have a much shorter refractory period, allowing them to undergo summation and reach maximum tension when repeatedly stimulated. In the left graph, we see that in a skeletal muscle fast twitch fiber, the refractory period, shown in yellow, is very brief compared to the time required for muscle contraction and relaxation. This short refractory period allows skeletal muscle fibers to be re-stimulated before the first contraction has fully ended. The right graph demonstrates the effects of repeated stimulation in skeletal muscle. If additional stimuli occur before the muscle has fully relaxed, tension begins to build. This process is known as summation, where contractions overlap and generate greater force. If stimuli continue at high frequency without relaxation, the muscle reaches a sustained contraction called tetanus. This ability to undergo summation and tetanus is useful for skeletal muscles, as it allows them to maintain prolonged contractions needed for activities such as posture and movement. However, in cardiac muscle, summation and tetanus must be prevented to ensure rhythmic pumping of blood and continuous circulation. The contrast between skeletal and cardiac muscle in terms of refractory periods highlights the specialized functions of each tissue type, ensuring that the heart beats consistently while skeletal muscles generate force as needed. The action potentials of myocardial autorhythmic cells differ from those of contractile cells, allowing them to serve as the heart's natural pacemakers. These specialized cells generate rhythmic electrical impulses without requiring external stimulation. Unlike contractile cells, autorhythmic cells have an unstable membrane potential known as the pacemaker potential. This gradual depolarization ensures that the heart continues to beat automatically. In step 1, when the membrane potential reaches approximately -60 millivolts, specialized channels called funny channels briefly open. These channels allow sodium ions to enter the cell while a small amount of potassium exits. This slow influx of sodium gradually depolarizes the sinoatrial node, or SA node, the heart's primary pacemaker. In step 2, as the membrane potential becomes more positive, more funny channels open, along with a set of voltage-gated calcium channels. The continued influx of sodium and calcium moves the membrane potential toward the threshold for an action potential. In step 3, once the membrane reaches threshold, a different set of calcium channels fully opens, allowing a rapid influx of calcium ions. This large calcium entry creates a steep depolarization, causing the action potential to fire. Finally, in step 4, at the peak of the action potential, calcium channels close and voltage-gated potassium channels open. This leads to the efflux of potassium ions, repolarizing the membrane and bringing it back to its starting state, ready for the next cycle. This continuous cycle of pacemaker potentials allows the heart to generate rhythmic electrical impulses, ensuring a steady heartbeat and coordinated contractions throughout the cardiac cycle. Autorhythmic cells of the heart generate spontaneous action potentials due to their unstable membrane potential, known as the pacemaker potential. This property allows the heart to beat independently of external signals. In the first graph on the left, we see that the pacemaker potential gradually becomes less negative over time. This slow depolarization continues until the membrane reaches threshold, at which point an action potential is triggered. This automatic depolarization ensures that the heart maintains a steady rhythm. The middle graph highlights the ion movements responsible for generating this action potential. Sodium and calcium ions enter the cell during depolarization, with calcium playing a major role in driving the membrane potential upward. As the action potential peaks, potassium ions exit the cell, leading to repolarization. The graph on the right shows how different ion channels behave during this process. At the beginning of the pacemaker potential, specialized funny channels open, allowing a slow influx of sodium. As depolarization progresses, calcium channels open, leading to a rapid influx of calcium and the generation of the action potential. At the peak, calcium channels close and potassium channels open, allowing potassium to exit and repolarize the cell. This rhythmic cycling of ion movement allows autorhythmic cells, particularly those in the sinoatrial node, to serve as the heart's natural pacemaker, ensuring a continuous and coordinated heartbeat. The heart's electrical conduction system ensures that each heartbeat is coordinated, allowing the atria and ventricles to contract in a synchronized manner. This system consists of specialized autorhythmic cells that generate and transmit electrical impulses throughout the heart. The process begins in the sinoatrial node, or SA node, located in the right atrium. The SA node acts as the heart's primary pacemaker, generating electrical impulses that spread through the internodal pathways to the left and right atria, causing them to contract. Next, the electrical signal reaches the atrioventricular node, or AV node. Here, conduction slows slightly, allowing the atria time to complete contraction before the ventricles are stimulated. From the AV node, the impulse travels down the atrioventricular bundle, also known as the bundle of His. This bundle splits into right and left bundle branches, which carry the electrical signal down through the interventricular septum toward the apex of the heart. Finally, the impulse reaches the Purkinje fibers, which rapidly distribute the signal throughout the ventricular myocardia. This causes the ventricles to contract from the bottom upward, efficiently pushing blood out of the heart and into the pulmonary and systemic circulations. This highly coordinated conduction system ensures that the heart beats in a rhythmic, efficient manner, maintaining proper blood flow to the lungs and body. The electrocardiogram, or ECG, is a diagnostic tool that records the summed electrical activity of the heart. Unlike a single action potential, which represents the electrical activity of an individual cardiac cell, the ECG provides a broader view of the coordinated electrical events occurring across all heart cells. The ECG consists of three main waves, each corresponding to a specific phase of the heart's electrical cycle. The P wave represents depolarization of the atria. This occurs as the sinoatrial node fires, initiating the contraction of the atria and the start of the cardiac cycle. The QRS complex represents ventricular depolarization. This is the most prominent wave on the ECG and corresponds to the rapid electrical activation of the ventricles, leading to their contraction. During this phase, atrial repolarization also occurs, but it is masked by the larger ventricular signal. The final wave, the T wave, represents ventricular repolarization. This occurs as the ventricles reset electrically, preparing for the next contraction cycle. By analyzing the shape, timing, and amplitude of these waves, healthcare professionals can assess heart rhythm, detect abnormalities, and diagnose various cardiac conditions. The electrocardiogram, or ECG, is divided into distinct waves, segments, and intervals that represent different phases of the heart's electrical activity. This recorded ECG tracing highlights the key components of a normal cardiac cycle. The P wave represents atrial depolarization, which occurs as the sinoatrial node fires and electrical activity spreads through the atria, leading to atrial contraction. The PR segment corresponds to conduction through the atrioventricular node and atrioventricular bundle. This brief delay ensures that the atria have time to contract before the electrical impulse reaches the ventricles. The QRS complex represents ventricular depolarization. This sharp, tall waveform indicates the rapid spread of electrical activity through the ventricles, triggering ventricular contraction. During this phase, atrial repolarization also occurs but is masked by the larger ventricular signal. The T wave represents ventricular repolarization. This is the phase where the ventricles recover electrically, preparing for the next contraction cycle. Segments and intervals provide additional diagnostic information. The PR interval measures the time from atrial depolarization to the start of ventricular depolarization, while the QT interval represents the total time for ventricular depolarization and repolarization. By analyzing these ECG components, healthcare providers can assess heart function, detect arrhythmias, and diagnose various cardiac conditions, making the ECG an essential tool in clinical practice. The electrocardiogram, or ECG, provides a recording of the summed electrical activity of all the heart cells as measured from the surface of the body. This allows healthcare professionals to assess the heart's rhythm and electrical conduction. In the first diagram on the left, we see a typical ECG waveform. Each peak and segment represents different phases of cardiac electrical activity, from atrial depolarization to ventricular repolarization. The middle diagram illustrates Einthoven's triangle, a system used to measure ECG recordings. Electrodes are placed on both arms and one leg, forming a triangular arrangement. Each pair of electrodes creates a different electrical lead, with one serving as the positive electrode and the other as the negative electrode. By recording electrical activity from different leads, clinicians can analyze the direction and strength of the heart's electrical impulses. The diagram on the right explains how the heart's electrical activity can be represented by a vector arrow, which shows the overall direction of current flow during atrial depolarization. The sinoatrial node, or SA node, initiates the electrical impulse, which then spreads toward the atrioventricular node and throughout the atria, generating the P wave seen on the ECG. Together, these concepts provide a foundation for understanding how ECG recordings reflect the heart's electrical function and how they are used to diagnose cardiac conditions. The electrocardiogram, or ECG, measures the electrical activity of the heart using electrodes placed on the skin. The deflection or movement of the ECG trace depends on the relationship between the direction of the electrical current and the axis of the lead measuring it. In the first diagram on the top left, an upward deflection on the ECG occurs when the electrical current moves toward the positive electrode. This indicates depolarization in the direction of the lead, producing a positive wave on the ECG tracing. In the middle diagram, a downward deflection occurs when the electrical current moves toward the negative electrode. This results in a negative wave on the ECG tracing. The third diagram shows that if the electrical vector is perpendicular to the axis of the electrode, no deflection occurs, and the ECG remains at baseline. This means there is no net movement toward or away from the electrode. The bottom diagram compares the ECG tracing to a single cardiac action potential. The action potential of a ventricular cell is an intracellular recording, where one electrode is placed inside the cell and the other is placed outside. Unlike the ECG, which measures the summed electrical activity of the heart, the action potential represents the voltage change within a single cardiac cell. In both cases, an upward deflection corresponds to depolarization, while a downward deflection represents repolarization. However, the action potential has a much greater amplitude because it is recorded closer to the electrical source. Understanding these principles helps interpret ECG readings and provides insights into the electrical conduction and overall function of the heart. Electrocardiograms, or ECGs, provide valuable insight into the electrical activity of the heart. This slide presents examples of both normal and abnormal ECG recordings, each representing 10-second intervals. The first tracing at the top left shows a normal ECG. It features a consistent pattern of P waves, QRS complexes, and T waves, indicating regular atrial and ventricular activity with a normal heart rate and rhythm. The second tracing represents a third-degree heart block, a condition in which the electrical signals between the atria and ventricles are completely disrupted. The P waves occur at a regular rate, but they are not followed by QRS complexes, indicating that the atria and ventricles are beating independently of each other. The third tracing shows atrial fibrillation, an irregular rhythm caused by rapid, uncoordinated electrical activity in the atria. This results in an absence of distinct P waves and an irregular spacing between QRS complexes, leading to an erratic heart rhythm. The fourth tracing represents ventricular fibrillation, a life-threatening arrhythmia where the ventricles quiver instead of contracting properly. There are no organized QRS complexes, and the electrical activity appears chaotic. This condition requires immediate medical intervention, such as defibrillation, to restore normal rhythm. The ECG analysis box provides key questions to assess heart rhythm. It prompts clinicians to examine heart rate, rhythm regularity, and the relationship between P waves and QRS complexes. Identifying discrepancies in these patterns helps diagnose arrhythmias and other cardiac abnormalities. By analyzing ECG waveforms and identifying deviations from normal patterns, healthcare providers can detect and manage various heart conditions, ensuring timely and effective treatment. This diagram illustrates the correlation between an electrocardiogram tracing and the electrical events occurring in the heart during a single cardiac cycle. The depolarizing regions are shown in purple, while repolarizing regions are highlighted in peach. The cycle begins with the P wave, which represents atrial depolarization. This electrical activity originates from the sinoatrial node, causing the atria to contract and push blood into the ventricles. Next, the PQ segment, also called the PR segment, represents the conduction of electrical signals through the atrioventricular node and the atrioventricular bundle. This brief delay ensures that the ventricles have enough time to fill before contraction. Following this, the Q wave marks the beginning of ventricular depolarization as the signal moves through the interventricular septum. The R wave represents the main phase of ventricular depolarization, where the signal spreads rapidly through the Purkinje fibers, triggering a strong contraction of the ventricles. The S wave marks the final phase of ventricular depolarization as the electrical impulse spreads upward from the base of the heart. After ventricular contraction, the T wave represents ventricular repolarization as the heart muscle resets and prepares for the next beat. By analyzing the relationship between ECG waveforms and cardiac electrical events, clinicians can assess heart function, detect conduction abnormalities, and diagnose arrhythmias, ensuring accurate and effective cardiac care. The cardiac cycle refers to the sequence of events that occur during one complete heartbeat, involving periods of relaxation and contraction of the heart muscle. During diastole, the cardiac muscle relaxes, allowing the heart chambers to fill with blood. In contrast, during systole, the cardiac muscle contracts, propelling blood into circulation. The cardiac cycle consists of 5 phases, beginning with the heart at rest. In the first phase, known as atrial and ventricular diastole, the atria are filling with blood from the superior vena cava, inferior vena cava, coronary sinus, and pulmonary veins. The atrioventricular valves remain open, allowing blood to flow passively into the ventricles. The second phase is the completion of ventricular filling, known as atrial systole. At this stage, the atria contract at the end of ventricular diastole, pushing a small additional volume of blood into the ventricles to ensure they are fully filled. The volume of blood in the ventricles at the end of this phase is known as the end diastolic volume. This represents the maximum amount of blood the ventricles hold before contraction begins. This structured cycle of filling and contraction allows the heart to function as an efficient pump, maintaining continuous blood flow to the body and lungs. As the cardiac cycle progresses, the heart transitions from ventricular filling to ventricular contraction, generating the first heart sound and initiating blood ejection. In the third phase, known as early ventricular contraction, the atrioventricular valves close, preventing backflow of blood into the atria. The vibrations that result from this valve closure produce the first heart sound, commonly heard as lub during auscultation. At this stage, no blood enters or exits the ventricles, a phase known as isovolumic ventricular contraction. However, as the ventricular muscle contracts, pressure within the ventricles begins to rise. Concurrently, the atria enter diastole, relaxing and passively filling with blood in preparation for the next cycle. In the fourth phase, known as ventricular systole, the ventricles contract forcefully. As ventricular pressure exceeds the pressure in the arteries, the semilunar valves open, allowing blood to be ejected into the pulmonary and systemic circulations. Following ejection, a small amount of blood remains in the ventricles. This is known as the end systolic volume, or the volume of blood left in the ventricles after contraction. These carefully timed events ensure that the heart functions as a powerful, coordinated pump, maintaining continuous circulation throughout the body. The final phase of the cardiac cycle is ventricular relaxation, which prepares the heart for the next beat and completes one full cycle. As blood in the arteries begins to flow back toward the heart, the semilunar valves close, preventing backflow into the ventricles. The vibrations from this valve closure produce the second heart sound, commonly heard as dup during auscultation. With the semilunar valves closed, the ventricular muscles relax, and ventricular pressure begins to drop. However, during this phase, ventricular pressure still remains slightly higher than atrial pressure. This period is known as isovolumic ventricular relaxation, where no blood enters or exits the ventricles. The heart remains in this state until ventricular pressure drops below atrial pressure. Once ventricular pressure falls below atrial pressure, the atrioventricular valves open, allowing blood to flow from the atria into the ventricles, marking the beginning of the next cardiac cycle. This rhythmic sequence of contraction and relaxation ensures efficient blood circulation and maintains the heart's continuous function as a pump. Stroke volume refers to the amount of blood ejected by one ventricle during a single contraction. It is calculated as the difference between the end diastolic volume, which is the blood volume before contraction, and the end systolic volume, which is the blood volume remaining after contraction. On average, the stroke volume for a healthy individual weighing 70 kilograms at rest is approximately 70 milliliters per beat. Cardiac output is an essential measure of heart performance, representing the total volume of blood pumped by one ventricle per minute. It is calculated using the formula: cardiac output equal heart rate multiplied stroke volume. For an average adult, cardiac output is approximately 5 liters per minute, ensuring adequate blood circulation to meet the body's oxygen and nutrient demands. Understanding stroke volume and cardiac output is essential for assessing heart function and diagnosing cardiovascular conditions. Cardiac output is the total volume of blood pumped by the heart per minute and is determined by two key factors: heart rate and stroke volume. Heart rate is controlled by the rate of depolarization in autorhythmic cells. This rate can either decrease due to parasympathetic innervation or increase in response to sympathetic stimulation and the release of epinephrine. When sympathetic activation occurs, the heart beats faster, increasing cardiac output. Stroke volume refers to the amount of blood ejected by the ventricles with each contraction and is determined by the force of contraction in the ventricular myocardia. This force is influenced by contractility, which is enhanced by sympathetic stimulation and epinephrine, and by end diastolic volume, the amount of blood in the ventricles before contraction. End diastolic volume is affected by venous return, which is the amount of blood returning to the heart. Venous return increases through venous constriction and is further aided by the skeletal muscle pump and the respiratory pump. The skeletal muscle pump works by compressing veins during movement, pushing blood toward the heart, while the respiratory pump enhances blood flow during breathing. By adjusting heart rate and stroke volume, the body regulates cardiac output to meet changing oxygen and metabolic demands, ensuring that tissues receive adequate blood supply under varying conditions. The autonomic nervous system plays a crucial role in regulating heart rate by altering the pacemaker potential of the sinoatrial node. It does this through two opposing mechanisms: parasympathetic control and sympathetic control. Parasympathetic control slows the heart rate by acting on muscarinic receptors in the sinoatrial node. This leads to an increase in potassium permeability, which causes the pacemaker potential to begin at a lower, more negative value. At the same time, calcium permeability decreases, which slows the rate of depolarization. As a result, it takes longer for the membrane potential to reach the threshold needed to generate an action potential, effectively slowing the heart rate. In contrast, sympathetic control increases the heart rate by stimulating beta-1 adrenergic receptors in the sinoatrial node. This enhances sodium and calcium permeability, increasing the rate of pacemaker depolarization. Because the membrane potential reaches threshold more quickly, action potentials fire at a faster rate, leading to an increased heart rate. Through these mechanisms, the autonomic nervous system dynamically adjusts heart rate to meet the body's changing demands, such as during rest, exercise, or stress. The autonomic nervous system regulates heart rate by influencing the pacemaker potential of autorhythmic cells in the sinoatrial node. This regulation occurs through parasympathetic and sympathetic stimulation, each affecting the membrane potential differently. In the graph on the left, parasympathetic stimulation hyperpolarizes the membrane potential of the autorhythmic cells, making it more negative. This increase in potassium permeability causes the pacemaker potential to start at a lower voltage, slowing the rate of depolarization. As a result, the membrane takes longer to reach threshold, leading to fewer action potentials and a decreased heart rate. This slower rhythm conserves energy and is more dominant during periods of rest. In contrast, the graph on the right illustrates the effects of sympathetic stimulation. Here, epinephrine and norepinephrine act on beta-1 adrenergic receptors, increasing sodium and calcium permeability. This leads to a more rapid depolarization of the pacemaker potential, allowing the membrane to reach threshold faster. With shorter intervals between action potentials, heart rate increases, enabling the body to respond to increased metabolic demands, such as during exercise or stress. By modulating the pacemaker potential, the autonomic nervous system ensures that heart rate is adjusted appropriately to maintain homeostasis and meet the body's ever-changing physiological needs. The cardiovascular control center, located in the medulla oblongata, plays a crucial role in regulating heart rate by coordinating autonomic input from both the parasympathetic and sympathetic nervous systems. This diagram illustrates how these two systems exert antagonistic control over the sinoatrial node to modulate heart rate. On the left side, parasympathetic neurons release acetylcholine, which binds to muscarinic receptors on autorhythmic cells in the sinoatrial node. This triggers an increase in potassium efflux and a decrease in calcium influx, leading to hyperpolarization of the cell membrane. As a result, the rate of depolarization slows down, delaying the threshold for action potentials. This process effectively reduces heart rate, promoting a state of relaxation and energy conservation. On the right side, sympathetic neurons release norepinephrine, which binds to beta-1 adrenergic receptors on the autorhythmic cells. This stimulates an increase in sodium and calcium influx, accelerating depolarization. With the threshold for action potentials being reached more quickly, heart rate increases, preparing the body for heightened activity, such as during exercise or stress. Through this dual control mechanism, the autonomic nervous system ensures that heart rate is continuously adjusted to meet the body's varying physiological demands, maintaining homeostasis and optimizing circulation. The autonomic nervous system regulates heart rate through two opposing mechanisms, parasympathetic and sympathetic stimulation, both acting on the sinoatrial node. On the left side, parasympathetic stimulation decreases heart rate. Acetylcholine is released by parasympathetic neurons and binds to muscarinic receptors on the sinoatrial node. This increases potassium efflux and decreases calcium influx, leading to hyperpolarization and a slower rate of depolarization. As a result, fewer action potentials are generated, reducing the number of heartbeats per second as shown in the electrocardiogram tracing. This parasympathetic influence is dominant during rest, promoting a lower, energy-efficient heart rate. On the right side, sympathetic stimulation increases heart rate. Norepinephrine binds to beta-1 adrenergic receptors on the sinoatrial node, increasing sodium and calcium influx. This leads to faster depolarization, allowing the membrane potential to reach threshold more quickly. The result is a higher frequency of action potentials, increasing the number of heartbeats per second, as seen in the electrocardiogram tracing. This sympathetic activation is crucial during exercise, stress, or emergencies, preparing the body for increased metabolic demands. Together, these mechanisms allow the autonomic nervous system to dynamically regulate heart rate based on the body's needs, ensuring proper circulation and maintaining homeostasis.